Mendel's low-profile existence as monk, teacher, researcher ended abruptly in 1868, when, on Knapp's death, he was elected abbot of the monastery. Although he continued his research, increasingly on bees and the weather, administrative duties were a burden, especially as the monastery became embroiled in a messy dispute over back taxes. Other factors, too, hampered him as a scientist. Portliness eventually curtailed his fieldwork. As he wrote, hill climbing had become very difficult for me in a world where universal gravitation prevails. His doctors prescribed tobacco to keep his weight in check, and he obliged them by smoking 20 cigars a day, as many as Winston Churchill. It was not his lungs, however, that let him down. In 1884, at the age of 61, Mendel succumbed to a combination of heart and kidney disease. Not only were Mendel's results buried in an obscure journal, but they would have been unintelligible to most scientists of the era. He was far ahead of his time, with his combination of careful experiment and sophisticated quantitative analysis. Little wonder, perhaps, that it was not until 1900 that the scientific community caught up with him. The rediscovery of Mendel's work by 3 plant geneticists interested in similar problems provoked a revolution in biology. At last, the scientific world was ready for the monk's peas. Mendel realized that there are specific factors, later to be called genes, that are passed from parent to offspring. He worked out that these factors come in pairs, and that the offspring receives one from each parent. Noticing that peas came in 2 distinct colors, green and yellow, he deduced that there were 2 versions of the pea color gene. A pea has to have 2 copies of the G version if it is to become green, in which case we say that it is GG for the pea-colored gene. It must therefore have received a G pea-color gene from both of its parents. However, yellow peas can result both from YY and YG combinations. Having only 1 copy of the Y version is sufficient to produce yellow peas. Y trumps G. Because in the YG case, the Y signal dominates the G signal, we call Y dominant. The subordinate G version of the pea-color gene is called recessive. Each parent pea plant has 2 copies of the pea-color gene, yet it contributes only 1 copy to each offspring. The other copy is furnished by the other parent. In plants, pollen grains contain sperm cells, the male contribution to the next generation, and each sperm cell contains just 1 copy of the pea-color gene. A parent pea plant with a YG combination will produce sperm that contain either a Y version or a G one. Mendel discovered that the process is random. 50% of the sperm produced by that plant will have a Y and 50% will have a G. Suddenly, many of the mysteries of heredity made sense. Characteristics like the Habsburg lip that are transmitted with a high probability, actually 50%, from generation to generation are dominant. Other characteristics that appear in family trees much more sporadically, often skipping generations, may be recessive. When a gene is recessive, an individual has to have 2 copies of it for the corresponding trait to be expressed. Those with 1 copy of the gene are carriers. They don't themselves exhibit the characteristic, but they can pass the gene on. Albinism, in which the body fails to produce pigment so the skin and hair are strikingly white, is an example of a recessive characteristic that is transmitted in this way. Therefore, to be albino, you have to have 2 copies of the gene, 1 from each parent. This was the case with the Reverend Dr. William Archibald Spooner, who was also, perhaps only by coincidence, prone to a peculiar form of linguistic confusion whereby, for example, a well-oiled bicycle might become a well-boiled icicle. Such reversals would come to be called Spoonerisms in his honor. Your parents, meanwhile, may have shown no sign of the gene at all. If, as is often the case, each has only 1 copy, then they are both carriers. The trait has skipped at least one generation. Mendel's results implied that things, material objects, were transmitted from generation to generation. But what was the nature of these things? At about the time of Mendel's death in 1884, scientists using ever-improving optics to study the minute architecture of cells coined the term chromosome to describe the long, stringy bodies in the cell nucleus. But it was not until 1902 that Mendel and chromosomes came together. A medical student at Columbia University, Walter Sutton, realized that chromosomes had a lot in common with Mendel's mysterious factors. Studying grasshopper chromosomes, Sutton noticed that most of the time they were doubled up, just like Mendel's paired factors. But Sutton also identified one type of cell in which chromosomes were not paired, the sex cells. Grasshopper sperm have only a single set of chromosomes, not a double set. This was exactly what Mendel had described. His pea plant sperm cells also only carried a single copy of each of his factors. It was clear that Mendel's factors, now called genes, must be on the chromosomes. In Germany, Theodor Boveri independently came to the same conclusion as Sutton. And so the biological revolution their work had precipitated came to be called the Sutton-Boveri chromosome theory of inheritance. Suddenly, genes were real. They were on chromosomes, and you could actually see chromosomes through the microscope. Not everyone bought the Sutton-Boveri theory. One skeptic was Thomas Hunt Morgan, also at Columbia. Looking down the microscope at those stringy chromosomes, he could not see how they could account for all the changes that occur from one generation to the next. If all the genes were arranged along chromosomes, and all chromosomes were transmitted intact from one generation to the next, then surely many characteristics would be inherited together. But since empirical evidence showed this not to be the case, the chromosomal theory seemed insufficient to explain the variation observed in nature. Being an astute experimentalist, however, Morgan had an idea how he might resolve such discrepancies. He turned to the fruit fly, Drosophila melanogaster, the drab little beast that ever since Morgan has been so beloved by geneticists. In fact, Morgan was not the first to use the fruit fly in breeding experiments. That distinction belonged to a lab at Harvard that first put the critter to work in 1901. But it was Morgan's work that put the fly on the scientific map. Drosophila is a good choice for genetic experiments. It is easy to find, as anyone who has left out a bunch of overripe bananas during the summer well knows. It is easy to raise. Bananas will do as feed. And you can accommodate hundreds of flies in a single milk bottle. Morgan's students had no difficulty acquiring milk bottles, pinching them at dawn from doorsteps in their Manhattan neighborhood. And it breeds and breeds and breeds. A whole generation takes about 10 days, and each female lays several hundred eggs. Starting in 1907 in a famously squalid, cockroach-infested, banana-stinking lab that came to be known affectionately as the Fly Room, Morgan and his students, Morgan's boys as they were called, set to work on fruit flies. Unlike Mendel, who could rely on the variant strains isolated over the years by farmers and gardeners, yellow peas as opposed to green ones, wrinkled skin as opposed to smooth, Morgan had no menu of established genetic differences in the fruit fly to draw upon. And you cannot do genetics until you have isolated some distinct characteristics to track through the generations. Morgan's first goal, therefore, was to find mutants, the fruit fly equivalents of yellow or wrinkled peas. He was looking for genetic novelties, random variations that somehow simply appeared in the population,